Dear friends and my dear students, here we deliver my first live video presentations on the mechanism of actions, different types of mechanism of actions for different antibiotics. Firstly, we here clear about the mechanism of action of the sulfonamides. Many bacteria synthesize their own folic acids, of which para-aminobenzoic acid is a constituent, and is taken up from the medium. Woods and Fields 1940s proposed the hypothesis regarding sulfonamide action. I think sulfonamides, being structural analogues of PABA, inhibit bacterial folate synthetase, which are enzymes responsible for DNA synthesis in bacteria. Folic acid is not formed and a number of essential metabolic reactions suffers. Sulfonamides competitively inhibit the union of para-aminobenzoic acid with pteridine residue to form dihydrofolic acids. Dihydroteric acids, which conjugate with glutamic acid to produce dihydrofolic acids, also being chemicals similar to PABA. The sulfonamides may itself get incorporated to form an altered folate, which is metabolic injurious for bacterial growth. Firstly, I think PABA is converted into folic acid synthetase into dihydrofolic acids. Dihydrofolic acid in the presence of enzymes dihydrofolic acid synthetase tetrahydrofolic acid form. This tetrahydrofolic acid is the precursor of nucleosides. Okay, here sulfonamides inhibit the folic acid synthetase enzymes. Hence, the folic acid synthesis will be blocked and nucleosides of any bacterial growth inhibited, and transcription and translation will be stopped. Another, here we delivered another beta lactam antibiotics mechanism of actions. I know all beta lactam antibiotics interfere with synthesis of bacterial cell wall. The bacterial synthesize UDP N acetyl muramic acid, which are pentapeptides, and UDP N acetyl glucosamine. The peptidoglycan residue are linked together. The peptidoglycan residue are linked together, forming long chain of a long strands, and UDP is split off. The final step is cleavage of terminal D-alanine of the peptide chains by transpeptidase. The energy so released is utilized for establishment of cross-linkage between peptide chains of the neighboring strands. This cross-linking provides stability and rigidity to the cell wall. The beta-lactam antibiotics inhibit the transpeptidase so that cross-linking, which maintains the loose knit structure of the cell wall, does not take place, and hence these enzymes and related protein constitute the penicillin binding proteins, which have been located in the bacterial cell wall. In Hindi, सबसे पहले यूडीपी एन स्टाइल म्यूरेमिक एसिड एंड यूडीपी एसिड ग्लूकोसामाइन एक ऐसे ये क्या करते हैं, बैक्टीरिया की सेल वाल सिंथेसाइज करती है, जो कि एक दूसरे से लिंक कर कर उसमें से यूडीपी स्प्लिट ऑफ हो जाती है और आपस में क्लीवेज या एक चेन लाइक स्ट्रक्चर फॉर्म कर लेती है. डी एलिन ऑफ द पेप्टाइड चेन्स इन द प्रेजेंस ऑफ ट्रांसपेप्टाइडेज. इससे ह्यूज अमाउंट ऑफ एनर्जी रिलीज होती है, जो कि क्रॉस लिंकिंग के लिए इंपॉर्टेंट रोल प्ले करती है. पेप्टाइड चेन्स बनते हैं नेबरिंग स्ट्रैंड्स के. अब क्या करता है, बैक्टीरियल सेल वॉल जो होती है पेप्टाइडोग्लाइकॉन की बनी होती है, इस ट्रांसपेप्टाइडेज एंजाइम्स को बीटा लैक्टम एंटीबायोटिक की तरह इनहिबिट कर दिया जाता है, जिससे वो अपनी सेल वॉल की प्रोटीन सिंथेसिस नहीं बना पाती है और उसकी सेल वॉल जो निटिंग स्ट्रक्चर लाइक धागे को पोहने का स्ट्रक्चर्स निटिंग लाइक स्ट्रक्चर ऑफ द सेल वॉल वो क्या करता है, वो इनहिबिट हो जाता है, जिससे कि जो एक इससे एक प्रोटीन्स का फॉर्मेशन होता है. दिस एंजाइम से रिलेटेड प्रोटीन्स constitute the protein binding penicillin binding proteins which have been located in the bacterial cell membrane. Okay, next mechanism of actions is the mechanism of action of the chloramphenicol. The basically chloramphenicol inhibit bacterial protein synthesis by interfering with transfer of the elongated peptide chain to the newly attached amino acyl tRNA at the ribosome mRNA complex. It is specially attached to the 50S ribosomes and thus may hinder the access of amino acyl tRNA to the acceptor sites for amino acid incorporation. Probably by acting as a peptide analogs, it prevents formation of peptide bonds. At high dose, it can inhibit mammalian mitochondrial protein synthesis as well. Okay, here in in protein synthesis in Hindi, in protein synthesis there are two steps: chain elongation steps, and last one is the translation steps. There are in 70S ribosomes or in bacterial 50S ribosomes we here take place. There are ribosomes in bacteria. Ribosome have two units. First one is 50S and another one is 30S. There are two sites. One is P site and another one is A site. Here we incorporate the one diagram. Please see here. These are, you see here, first is 50S ribosome larger units and 30S smaller units. There are C3 C4 positions. Here P sites and another one is A sites, and in P sites receptors are present where A1 A2 A3 amino acids are attached, and in A sites here we in the presence of tRNA new amino acids A4 are attached at C4 positions. This is the amino acid transport chains. And secondary, these P sites and A sites peptide bond formation takes place. A1 A2 A3 are attached in the P sites and A4 is attached on the newly sites on A side. There are peptide bond formation takes place, and here in last third diagram there C3 sites are reopens and all amino acid chains are moved to the above the position of the A4. A1 A2 A3 and A4, these are ribosomes move to next mRNA codon, and next one is the translocation. In translocation here we see A1 A2 A3 and A4, all four amino acids, these are all four amino acids are transferred from the p sites and newly amino acids are coming for waiting for the A5 position into the amino acyl sites A sites, and peptide sites 1 2 3 4 amino acids are bound, and there is a chain is formation of the amino acids which finally form the protein bindings. These are steps of the chain elongation and translocation. Here we studies about the next mechanism of action for tetracycline. Here we see some mRNA attached to the 30S ribosomes. These second smaller units mRNA attached initiate complex of mRNA from volosome formation, nascent protein chain formation at 50S ribosomes. Next amino acid come to the A site or acceptor sites, peptide bond formation from new amino acids, elongated peptide chain shift from A site to the P site, ribosome move from mRNA to the next codon. Here is termination codon, nonsense codon, also known as nonsense codon, UUU, UCU, UUC. These are nonsense codons are takes place and process terminate protein synthesis. These are the tetracycline flow chart of mechanism of action of tetracycline. Last one is fifth is quinolones. The fluoroquinolones inhibit the enzyme bacterial DNA gyrase, which makes which nick double-stranded DNA introduce negative supercoils and then reseal the nicked end. This is necessary to prevent excessive positive supercoiling of the strands, which when they separate to permit the application of transcription. The DNA gyrase consists of two A and two B subunits. The A subunit carries out nicking of DNA. B subunits introduce negative supercoiling, and then A subunit reseal the strands. Fluoroquinolones bind to A subunits with high affinity and interfere with its strands cutting and resealing functions. Recently, evidence indicate that in gram-positive bacteria, the major target of fluoroquinolones action is a similar enzyme topoisomerase 4th. First enzyme is DNA gyrase and another one is topoisomerase 4th, which nicks and separate daughter DNA strands after DNA replications. Greater affinity for topoisomerase 4th may confirms high potency against gram-positive bacteria. The bactericidal action probably results from digestion of DNA by exonuclease, whose production is signaled by the damaged DNA in place of DNA gyrase, or second one is topoisomerase 4th. The mammalian cells possess an enzyme topoisomerase 2nd, which have which has very low affinity for fluoroquinolones, since it shows a low toxicity of the host cells. There are fluoroquinolones. It inhibits the both two enzymes DNA gyrase and topoisomerase 4th, which inhibit the nicking gram-positive bacteria, inhibit the supercoiling, and protein synthesis also inhibited here. Quinolones. And another mechanism of action is aminoglycosides. Here, mechanism of action, the aminoglycosides are bactericidal antibiotics. Aminoglycosides are bactericidal antibiotic. Bactericidal means side माने kill करना antibiotics. All having the same general patterns of action, which may be described in two main steps. Which steps? Two main steps. Step first, transport the aminoglycoside through the bacterial cell wall and cytoplasmic membrane. And step second is binding of ribosomes, resulting in inhibition of protein synthesis. Transports of aminoglycoside into bacteria is a multi-step process. They diffuse across the outer coat of gram-negative bacteria through porine channels. Which channels? Porine channels. Entry from the periplasmic space across the cytoplasmic membrane is carrier mediated, which is linked to the ETS chain, electron transport chain. Thus, penetration is dependent upon maintenance of a polarized membrane on oxygen dependent active process and high pH. Step second is it is binding of ribosome, resulting in inhibition of protein synthesis. Second one is the inside the bacterial cell, streptomycin bind to 30S ribosomes. Which ribosome? 30S ribosome, but other aminoglycoside bind to additional sites on 50S subunits, larger subunits, as well as to 30S 50S interface. Middle position 50S and 30S interface. They freeze initiation of protein synthesis, prevent polysome formation, and promote their disaggregation to monosomes, so that only one ribosome is attached to one strand of mRNA. Only one ribosome is attached to the only to each strand of mRNA. Binding of aminoglycoside to 30S 50S junction causes distortion of mRNA codon recognition, resulting in misreading of the genetic code. यानी कि जो genetic code जो read कर रहा होगा वो misread करने लगेगा. Misreading of the genetic code thus inhibits protein synthesis. And last one is seventh one is the erythromycin. Erythromycin mechanism of action. Erythromycin is bacteriostatics. इट इज़ नॉट बैक्टीरियोसाइडल, इट इज़ बैक्टीरियोस्टैटिक्स। स्टैटिक्स माने रोकना, प्रिवेंट करना, स्टेट करना। Bacteriostatic at low but cidal for certain bacteria, but in the case of certain bacteria uh, at high concentration, it is bactericidal, but in at its optimum condition, concentration or low concentration, it is a bacteriostatic in nature. Cidal action depend on the organism concerned and its rates of multiplication. It's depend upon the in which rates multiplication takes place. Erythromycins act by inhibiting bacterial protein synthesis. It combines with 50S ribosome some units and interfere with translocation. Dear students, we here we attach some diagram in your Microsoft Word papers. Here we see 50S units and second is the 30S units, and there is two steps. One is peptide chain prolongation and last one is a translocation process. In but erythromycin act by inhibiting bacterial protein synthesis by combining with 50S ribosome subunit and interfere with translocation steps. After peptide bond formation between the newly attached amino acids and the nascent peptide chain at the A site, the elongated peptide is translocated back to the that P sites, making the A site available for next amino acids. tRNA attachment. This is prevented by erythromycin, and the ribosomes fails to move along the mRNA to expose the next codon. As an indirect consequence, peptide chain may be prematurely terminated. As an indirect consequence, peptide chain may be prematurely terminated. Synthesis and larger protein is suppressed. Okay. ओके, थैंक यू सर, थैंक यू बेटा। इस 7 मैकेनिज्म ऑफ एक्शंस को आप लोग सही से सुनिए, रिपीट करके